Okay, so we're gonna go over the process of meiosis. So the white circles represent the PMAT1, black circles are PMAT2, yellow circles represent the nuclear envelope, these orange things represent centrioles, green is the spindle fibers, and the blue represents one type of chromosome and the purple represents the other type. So let's see. This circle represents prophase 1. In prophase 1, it starts by the chromosomes start forming from the chromatin, and they form the chromosomes with 2 chromatids each. And the red also represents the centromere. So in prophase 1 for meiosis, the chromosomes start crossing over with each other so that they transfer their different alleles to the other chromosome that the homologous pairs pair up with. Then we go over to metaphase 1, where the homologous pairs line up in the middle. In anaphase 1, the spindle fibers of the centrioles start pulling the homologous pairs apart from the middle. In telophase 1, the nuclear envelope starts forming around the homologous pairs that were pulled to each side, because 2 new cells are going to be formed. So this is before cytokinesis occurs. So they're still partially attached, but they're about to split off into 2 different cells. And the centrioles ended up retracting and leaving the homologous pairs alone for later. Here is, we're going to start with prophase 1, or prophase 2, where here is the cell membrane. And to start, we're going to have the nuclear envelope with the homologous pairs inside. Or no, not homologous pairs, the chromosomes at this point. Yeah, back here, these were no longer homologous pairs. I think I said they were, because they were split apart. So these are just chromosomes, to take back what I said earlier. So the chromosomes will be inside the nuclear envelope, and they're not gonna cross over like they did back here in prophase 1. And prophase 2, they're going to reform from the chromatin, which they reverted to in, I believe, telophase 1. So they're going to reform into chromosomes. There would be the centrioles here, and they're going to move over to, let's see, metaphase 2, and they will be spindle fibers are going to start attaching to the centromeres, and they're going to start pulling away in anaphase 2, which would look like this, because they were pulled away to each side of they would stay inside the cell membrane, but this is just to show they were pulled to separate sides. And after this, the spindle fibers are going to let go and they're going to start retracting a little bit. They're still there, but they will retract. And in telophase 2, the chromosomes with 1 chromatid at this point will start unraveling into the chromatin again. See, so at this point, this is telophase 2, where the cell is splitting again into 2 new cells with 2 chromosomes, for my example, 2 chromosomes left, 2 chromosomes in each, for my example, with 2 chromatids and 2 chromatids that were pulled apart. I believe that's it. Don't believe I'm forgetting anything. That's all this process of meiosis will, not for my example, but overall, it's gonna start with one either spermatocyte or oocyte, and it's gonna end up resulting in 4 sperm cells or 4 egg cells, depending on whether it is going to be male or female producing them. I believe that's all.